independent assortment is another way we get variation in sexual reproduction and then end up with largely varied offspring from the same combination of parents and the same original kind of genetic information. So what we see is during or at the point of metaphase 1 in meiosis, so we can have two examples of the first metaphase here. What we can see is obviously sister chromatids are going to align along the metaphase plane. So if we look at maternal and paternal sister chromatids and keep them separate by using a couple of different colors here, then what we might have is all 23 original chromosomes that were received from the father might be sort of symbolized in red here. So these are paternal. And then in blue we might have the maternal. So let's say, for example, that what occurred was all of the original father's chromosomes in an individual whose one of their cells is about to undergo meiosis to create more sex cells, all of the person's father's chromosomes, the paternal chromosomes lined up along one side of the cell, and the maternal chromosomes, now sister chromatids because the DNA is doubled, line up along another side. And then we saw our spindle fibers pull each of those to opposite poles of the cell. Then what we would end up with is, ready for metaphase 2, this kind of arrangement where all of the maternal and all of the paternal sister chromatids went to one cell or the other. So then when we have metaphase 2 occurring in meiosis, these sister chromatids would be split like so. So daughter cells would be split once more, and we would end up with haploid cells containing either all paternal chromosomes or all maternal chromosomes, and that would be our sex cells at the end. So that's one extremely unlikely possibility, is all of the paternal and maternal cells, sister chromatids are lined up on opposite sides of the cell at metaphase 1 in meiosis, were pulled apart to opposite poles and then pulled apart again to create sex cells that were exclusively made up of the person's father's DNA or mother's DNA without there being a mixture. But this is extremely, extremely unlikely because of something called independent assortment. So I'll change that, my spelling mistake over here, before we look at a second possibility for metaphase. What's far more likely to happen is, because we've got these 23 pairs of sister chromatids arranging themselves in metaphase, it's highly, extremely unlikely that what we see in our first example would occur. More likely what's going to happen is there's going to be mixtures of different paternal and maternal chromosomes on either side. So, for example, if we just use this combination of 4 sister chromatids, obviously remembering that this is happening with 23 pairs in a full set, so we're just using 4 as an example, we might get the sister chromatids lining up in this sort of fashion. So what we can see is, as we go through this process, this will drastically affect the haploid cells, the sex cells that we get at the end. So if we had this arrangement, so our sister chromatids arrange themselves in this kind of format, you can see that we're going to get varied sex cells and varied genetic information at the end. So finally, what we would end up here with is sex cells that are a combination of both maternal and paternal chromosomes. I'll just draw some arrows in just to make sure we're aware of the kind of flow of information here, like so. So what you can see is, if we compare the sex cells at the end of each of these possible combinations or arrangements of sister chromatids is that this independent assortment, so the randomness in which the way these sister chromatids are going to align themselves in preparation for division in metaphase, is going to affect whether we see things such as all maternal sister chromatids or all paternal sister chromatids being present in a sex cell, or different mixtures of both, like we see here. So because of the amount of chromosomes, the amount of sister chromatids, the 23 pairs, and the possible combinations that we can see, it's something like 60 trillion different possibilities or different arrangements we can get at metaphase 1, which is one of the big reasons behind why we see so much variation in the offspring of the two parents. So why we don't have identical children is because of this massive variation, which leads to highly different kids genetically. And independent assortment is one of the biggest factors behind that.